our first global summer school in biomedicine and biotechnology. It is my pleasure to welcome you today. I hope we are having students and colleagues, academics, visitors from many places in the world. And of course, this global summer school is the global summer school in biomedicine and biotechnology. It is my pleasure to welcome you today. I hope we are having students and colleagues, academics, visitors from many places in the world. And of course, this global summer school is the global summer school in biomedicine and biotechnology. Apologies, I think there might have been a problem with some echo or some microphone maybe open. Hopefully that'll be sorted out now and we can start with the global summer school. As I was saying, this is a virtual summer school given the challenging times we are facing, but we are very hopeful that in the future we can start running this as a hybrid or maybe face-to-face hosting in the different countries, and even more as we get a greater consortium participating in this collaboration. I'm going to share my screen now, if that's okay. I cannot start until the last screen is stop. Could I get access, please, to sharing the screen? Excellent, thank you. So hopefully you're all seeing my screen now. Global Summer School, bienvenidos, bienvenuti, welcome. Welcome to everybody. It is a real pleasure to be able to say hello and welcome to many, many students. And of course, we have 4 hosting institutions, but we are hoping that this global invite went way beyond these institutions. And we have students from other parts of the world, which we will try to find out later on. But just so you know, of course, the map of the world, we have 2 Mexican higher education institutions, 2 universities, Enes Juriquilla from UNAM and UPIBI from the Instituto Politécnico Nacional in Mexico. And we also have an Italian university, Università del Piemonte Orientale in Novara, even though there's other campus. And of course, Manchester Metropolitan, which is where I am based at in Manchester in the UK. But there'll be more information about later on. As you know, if you've had a look at the program, the difference with this summer school is that it's not only about the science. We really want you to get that experience, that cultural knowledge and experience that you would get if you were to travel to another country to participate in these events. We know that because of COVID-19, there are many travel restrictions, there are many sanitary measures, and we know that virtual events have been the most participated because we just cannot travel. Because of that, we wanted to put together a program among these 4 institutions to provide our students with some experience abroad, as virtual as it could and as safe as it could, because safety of our students and staff, of course, is the priority of all of us. But we thought that we'll give it a kick, a change. We'll have, of course, very interesting scientific meetings, but we also want you to learn more about the country that is hosting the day in the event. As you can see, we have 4 days. The first one is hosted by Manchester Metropolitan. Tomorrow, you will be hosted by Enes Juriquilla. On Wednesday, by UPO Novara in Italy. And the last day, again, another Mexican institution, but it is a different city. So we're hoping to see a different flavor of activities and cultural talks that you will get. At the very beginning of every day, you will have a cultural session where we are trying to show you what would you be experiencing if you were in that city for the day or for the week. That will be followed by 2 scientific talks. We will have a bit of a screen break, a comfort break. If you want to stretch, you need to top off your glass of water, or coffee or tea. And that will be followed for 2 more scientific talks every day just to be finished by another cultural session. And that would be what a conference dinner would be if you were in that city. We know that conference dinners bring very fond memories to students. When you remember, oh yes, that conference, of course you remember the conference dinner. So we want to share part of that experience with you, and hopefully you'll agree it's a very enriching experience. Just at the end of every day, we're going to run a bit of a feedback survey. We want you to tell us, what did you enjoy, what you didn't so much, what was your preferred part, and of course we want to know a little bit more about you. So which country are you from, some details on your course that will help us to plan even a better summer school for next year, if that is possible. So without further ado, I will start. Actually, I will do the first talk of today, which is the cultural session. What could it be like to be in Manchester for this course? Now, the first question is, where are you going? Are you going to England? Are you going to the UK? Where is Manchester? And unless you're very good in your history and your geography, if you're not from the UK, it can be a bit confusing. And that is because actually the United Kingdom of Great Britain and Northern Ireland is a union of 4 nations: England, Scotland, Wales, and Northern Ireland. And you can see these 4 nations are part of Great Britain. Of course, each nation have their own flag, their own football team. Of course, if you're following the Euro Championship, you know that there are different teams for them. But we all come together under Great Britain, and that is why we have the Union Flag Jack, which is the one that incorporates most of the flags, showing that union. And in Olympics, for example, you might see Team GB, which is Team Great Britain. So it's not difficult to get confused whether it's Great Britain or England. But Manchester, as you can see here at the star, it's in the middle, the heart of the Great Britain, only 2 hours from train to London. Very close to Wales as well, to Northern Ireland and Ireland, if you want to visit the Titanic museum, or even Scotland if you want to go to Edinburgh or Glasgow. So location-wise is perfect when you come to Manchester to visit us or to attend a summer school or a course. This is the Mancunian, which is the word from someone from Manchester, the Mancunian coat of arms. But of course, Manchester is better represented by the working bee. And that is something similar you might see in other cities where there are big statues of animals that represents the city. In the case of Manchester, that's the working bee. And that is because Manchester is the birthplace of the Industrial Revolution. So we work really hard. And that is why we have the bee as a symbol. Now, when you travel, most of you would have arrived by a plane. You would have flew into Manchester. You would arrive to Manchester Airport. And you would be starting to see a lot of these hexagon figures. And that is because of 2 main reasons, really, really well told and joined together. Of course, as I said, it's all about the bees, working bees. And you know that this is the shape of a hive. But also, you might know that Manchester is hosted the 2 researchers, the Nobel Prizes that discovered graphene. And again, being carbon is one of the representations that we have. So that you will see a lot of hexagon figures along the city. Once you arrive to the airport, you can travel into the city by different transport methods. You can take the tram, you can take the train, you can take a bus, a taxi, not a ship. We do have canals, but not one that connects the airport to the city, but there are canals around Manchester, exactly because of that, the relevance of the Industrial Revolution and how products were transported in those days. Now, could you get lost in the city? Manchester is a big city for UK standards, not a big city for Mexican standards, of course, or even for Italian standards. But we have a pretty good total population of 3.2 million, I think was the last count, in Greater Manchester. Of course, when you arrive, probably you will be concentrated in the city center. The universities are in the city center. Most of the services are in the city center. Of course, there are lots of things to do around, but you will not be getting lost because everything is within walking distance. Now, when you arrive to the UK, the language is English, and we are very spoiled because we know that everybody who's not from the UK is joining this summer school because you are quite good in English. You can understand, you can speak, you can read in English, and that makes possible for us to run this whole summer school in English. So thank you very much for being so clever at languages and putting the effort to learn another language. We cannot say, unfortunately, that of everybody. But within the English language, of course, maybe some of you have learned the American English. Maybe you're more exposed to Hollywood movies or sitcoms. There are certain differences with the British English. One of the things, well, it's not a difference, but you have to watch your P's and Q's. And that means that you also have to say please and thank you. That's a very good thing about the British culture. It's always please and thank you. Now, you might hear cheers. If you are familiar with, again, with the TV program, you might think cheers is when you have an alcoholic drink and you are saying salud or chin chin, and you are, that's cheers, which you can use it. But cheers also means thank you. So if someone opens for you the door, you can say cheers, right? Something else you might not know, some people in the row might, on the street might say, oh, hiya doc, or hiya love, hiya hon, hiya mate. Hiya doc, hiya love doesn't mean that they're falling in love with you. It's just another word to say mate or person or friend or individual. So that could be quite regional as well. It's not necessarily you're going to have in Manchester all of these, but you could get them. Another one, tea is not tea, and that is just to confuse you. Tea, of course, is the drink that you can have, a hot drink most of the times, but tea is also used to describe supper or dinner. So it's also a meal, and they can ask you, oh, what are you having for tea? They're not asking you if you're having peppermint or chamomile. They're asking you if you're having, we'll go into more options for tea, but it's more about the meal. Queue, you might not be familiar with the word queue, or you might know the queue, but once you come to the UK, you know that you have to queue. You have to form an orderly line in order to gain access to products or services. That's something that actually has helped with vaccination in the UK. And quid is another word that, again, is not as common if you are not in the UK, which means pound. It means the currency that we use in the UK. So if you ask, how much is this mug? They might say, oh, 5 quids, which means £5. Okay, well, I did say we're going to talk a little bit about food, so you know more about the British food. And if you have watched movies, you might have seen the full English breakfast, which is a very hearty portion of fried eggs, baked beans, which is beans in a tomato sauce, mushrooms, bacon, sausage, grilled tomato, toast, and black pudding sometimes. Black pudding is clotted blood, something similar to moronga in Mexico. So this is the traditional British full English breakfast that you will get in your hotels the following morning after you flew in. Of course, not everybody has this every morning. So the usual, you would see British people having toast, tea, coffee, maybe some kind of roll, pastry, but it's not a very hearty breakfast as could be in Mexico, actually. Then, if you have a lot of time and you're celebrating something, you might go for an afternoon tea. An afternoon tea is when you have, you sit in the afternoon to have some tea, but that tea is going to go along with some sandwiches. And again, there are some stereotypes about cucumber sandwiches. And yes, it is bread, butter, cucumber, and breaded butter bread as well again. So you could have different sandwiches, coronation chicken, there are different flavors. Then you have some really nice small cakes and some pastries. And the very typical that you have is going to be a scone or a scone, depending where you're from. A scone is something very similar to the Mexican biscuit that you can halve it and put some clotted cream and jam, and you eat it with your tea or your coffee. That would be an afternoon tea. Of course, for lunch, we have the very famous fish and chips. And if you are in the north part of the country, you might as well have some curry. People in some regions like to add curry, which is an Indian spicy, delicious sauce, into the chips. Also, on a Sunday, you can have a Sunday roast, which is one of the typical lunches. You have some roast meat, in this case is chicken, You have a Yorkshire pudding, which is a delicious bun, which is very, very tasty. You have roasted potatoes, you have mash, peas, and a lot of gravy as well to help to go. But again, this is not what everybody would have here every day. So don't expect everybody will go for fish and chips every day. What usually you have for lunch, depending on your eating habits, sandwich, crisp, a chocolate, a buttered chocolate and soda, or you might have a soup, also depending on the weather. If it's a bit cold, you might want something to warm you up, or you might have some salads, but it's rather a light lunch, and it's around anything between 12 and 1, you would be having a lunch. So it's very early on the day compared to other lunches. In the UK, we are very fortunate to have influence from world cuisines, and the Indian and the Chinese cuisine are the ones that you can find the most. They are delicious cuisines, but you can also find Mexican and Italian. Now, a word of advice, don't try it. If you're coming to Manchester, what they understand for Mexican food is a burrito. That is almost as much as it extends in the knowledge. And for in terms of Italian food, I've been told that pasta and pizza are nowhere what it should be. So, of course, if you come to another country, you want to try what is local. You don't want to go. But we do know that sometimes, especially if you're here for a longer period of time, you might be tempted into some comfort home food. Don't. Now, pubs, English pubs, British pubs are also very famous. We have very traditional pubs, and you might have heard about the Guinness beer. There are different beers, whether it's an ale or a lager, what you have, or cider. In the UK, you also have very good ciders with different flavors, of course, based on apples, but they add all sorts of flavors. If you were to go to a pub, the piece of advice is just to know two things you need to know, and that was before COVID, of course. One is that when you sit at a table, you will find a number in that table. That's the number of your table. So knowing that number, you go to the bar, to the counter, and you say the number of your table and you order, and then you pay, go back, and you get the drinks to your table. The second thing you need to know is the measures to order beer or any drink. And here you will have a pint, so a full pint of liquid, or a half a pint, which you just ask for a half, which, especially if you're trying to taste a lot of different drinks, you might want to get started with halves instead of full pints for that. Sports. I don't want to assume anything, but I'm pretty sure that at least one of you know about the football clubs that we have in Manchester. Of course, Manchester City, Manchester United, big rivalry, but great for sports, a great city for sports. And if you're coming to Manchester, you might already have a favorite team, and you might want to go to that stadium, but we also have the National Football Museum, and these are 3 levels of activities and history based on football. I know that from many friends that have visited and colleagues, that's a must visit if they enjoy football. They have a great day there. But it's not the only sports that we have in Manchester. We have a velodrome, we have a tennis center, we have, of course, cricket, field hockey, and rugby. And if you're interested in any of these sports, you can either watch a match or even you can join if you were to come and move here, of course. Now, in terms of culture, I want to say we are as good as London, but I will be lying. But we do have a lot of very interesting and great venues. And we can start with museums. You can see the Museum of Manchester. We have the Science and Industry Museum. We have the Museum of the War. We have many galleries as well, art galleries, especially the Lowry Gallery, where we have exhibitions about Lowry paintings. We have the People's History Museum, and something that is very important, even though it's a very small house, it's not a huge museum, but it's the house of Emmeline Pankhurst. And if you don't know Emmeline Pankhurst, this is one of the main suffragettes, one of the main ladies that fought for women to be able to vote, to have the right to vote. And this is very celebrated in Manchester. We're very proud of having Emmeline Pankhurst as part of our heritage, but also we think that considering the issues of equality, it's something that unfortunately it needs to be reminded still. Now, if you're more into music, again, Manchester is your city. And we don't have enough time to tell you all the fabulous concerts and gigs that we have. But regardless, if you're more inclined towards classical music, or if you're more into pop, rock, metal, any kind of music, you will find a great venues to listen to the latest. So regardless, if you want to listen, well, Adele is out of circulation for the moment, but Ed Sheeran, or if you want to listen to Nikki Benedetti, you can go to any of the venues to listen to them. And of course, shows, as I did say, we do not have as many venues as London. We don't have the full West End, but we do have a couple of very good theatres that put the same shows that you would watch in London. So Opera House, Palace Theatre, Royal Exchange, and our own university, Manchester Metropolitan, has a state-of-the-art theatre, not only for our students' productions, but also to host other productions touring in the country and in the world. Manchester is a very inclusive city. It doesn't matter your ethnicity, your ethnical background, your religious background. It doesn't matter your gender identity. We are very open to all having a space and having a voice in our city. But of course, you might want to go outside. We also have near Manchester, there are great places to go and enjoy of the countryside, have a good hike. We do not have mountains like in Italy and in Mexico. What we call mountains, you would think they're hills, but still there are beautiful landscapes that you can enjoy, especially on a dry day. And in town, there are several things you can do when you take some hours out of your summer school course or your course. You can go to the Manchester Cathedral, you can go to the library, you can go to Arndale, which is the largest mall in town near the universities, or of course you can go to our official Manchester souvenir and news shop, where you can find anything and everything saying Manchester, from a mug to a t-shirt and even baby clothes would say in Manchester. So just to finalize this first cultural session, I hope I gave you a good flavor of what would you be expecting in Manchester and experiencing. This is the map of our university, Manchester Metropolitan. You can see the different buildings across the blocks. We have invested a lot of millions of pounds in new buildings to make sure that everything is a state of the art. And thank you very much for joining us on this first talk. I hope you had a good time, and we are going to continue with the program. Thank you.
Thank you very much, Araida, for this very nice introduction to Manchester and to the city, to the food, to the activities that we can find there. We're all ready to travel. So thank you. At least through YouTube and Zoom, we can do that now. So now we're going to continue with our program, and I'm going to introduce Dr. Jim Pritchett. He will give us a talk entitled Decoding Mechanosensing During Fibrotic Disease. Dr. Pritchett completed a PhD, in developmental neuroscience at the University of Manchester. And then he did a postdoc in vascular biology and organ fibrosis. His main interest now is investigating the role of mechanosignaling in fibrosis and the phenotypic switch that hepatic stellated cells make in response to liver injury. So please welcome Dr. Pritchett.
Hi everybody, I'm hoping everyone can hear me okay and my volume is at the right level.
Yeah, it's fine.
For the invitation. So yeah, hola, ciao, hello, whatever's appropriate. Thank you very much for the introduction. I'm going to try and be a little bit interactive if that's okay once I've introduced myself. So if people have a second screen or a device they can use, they could type in the web address menti.com. I'll put that on screen in a second. And hopefully there'll be a chance for you to interact a little bit with my talk as I go along. So yeah, that's that web address. So menti.com should take you to a website, and in a little while there will be a number that pops up. Just while I get used to using Zoom again, just bear with me. It's been a while. I've been on Teams for a long time. So if I present this, hopefully you can now see my Menti screen with the same greeting displayed, and the code at the top should let you into my presentation. So I'll give that a little bit of time while people log into that. And then the first question, just to check it's working, is just to ask which country people are actually in whilst I'm doing this presentation, which hopefully will get the ball rolling. And then after that, some of the questions become a little more scientific. So which country are you watching my presentation from? And hopefully we'll start to see some results coming in shortly in the form of a word cloud. So at least it's working for at least one person, which is a promising sign. So let's see if we have any other entries. Mexico is obviously the popular answer so far. Do we have any Italian entrants today? Okay, it's good to know that it's working at least, and we do have people from different places. So I'm going to switch back to my PowerPoint, but I'll come back to Menti shortly. So, as Magda kindly introduced, I'm going to talk about mechanobiology and a little bit about fibrosis. So, obviously, my name is Jim. I'm based at Manchester Met, which Araida just did an excellent job of introducing. And I'm really interested in one organ in particular, the liver. And this is a piece of that liver tissue stained histologically with a stain called pipistrellus red. And this is what normal healthy liver looks like. And the main thing to take from this is there's very little red staining. So this red staining is for collagen. And that's in places you'd expect to see it in a healthy liver. So around the vessels that go through that particular tissue. As a result of injury, which can be from any type of insult, the liver can become inflamed, and eventually this leads to cirrhosis, which is kind of the end stage of liver fibrosis, and it's thought to be largely irreversible. And in that scarred tissue that has gone through the process of fibrosis all the way to cirrhosis, you see much more of this red collagen staining. So the collagen is the protein that makes up the fibrotic scar in that fibrosed organ. And my research that I'm going to talk about is really about understanding a phenotypic switch that occurs in a type of cell called hepatic stellate cells. So these are the cells that we know drive most of the deposition of the extracellular matrix, including collagen, that makes up that fibrotic scar. And so what we're really interested in, as a basic scientist, I'm mainly interested in what goes on inside these cells to allow that change in phenotype. From a healthy hepatic stellate cell in a normal liver that is fairly lipid rich, stores things like vitamin A, which then changes its phenotype in response to that inflammatory insult to become something that can migrate. It proliferates more rapidly. It can migrate through the tissue, secreting the extracellular matrix proteins that keep the organ intact following injury. But over time, much like if you pick a scab on your skin, that deposition of fibrous material causes a scar, which disrupts how the organ should function normally. So after an acute injury, these cells are useful. They do something important. They maintain the architecture of the tissue, which will allow a useful healing response to occur and regeneration to continue. But if that inflammation occurs over time, as I say, it's like picking a scab. You end up with a scar in that particular organ, in this case, the liver, and that causes obviously issues around liver function and eventually cirrhosis, and ultimately can lead to death. So if we go back to menti.com, hopefully my windows are switching around appropriately. We can see we do have some entrants from Italy, which is good. So thank you. I'm from the UK and Mexico as well. So my next question is not something I'm expecting anyone particularly to have the correct answer for, because it certainly surprised me. But there's a statistic that gets cited quite widely about how many % of deaths, at least in the US, according to the US government, involve some kind of fibrosis. So that can be liver fibrosis, kidney fibrosis, scarring of the lungs, or other internal organs. So what percentage of deaths do we think the US government think involves some kind of fibrosis of any organ or tissue in particular? And this is on a scale of 0 to 100%. So if you went to 10, you're saying that all deaths involve fibrosis. If you went to 0, you're suggesting that no deaths at all involve fibrosis. So I'll let that run for a moment or so. It turns out you're all very well informed, because you're obviously better scientists than I was, because this answer is coming out around about the number that the US government has actually placed on this. So your 4.6 is very, very close to the 45% that the US government seems to suggest is the number of deaths that have fibrosis as some kind of cause or can be attributed in some way to a fibrotic process, which surprised me, because I thought half of all deaths almost was a very high number. But I guess when you start to think about the organs that can be affected, perhaps it's not that surprising, but still, it does seem a considerable number. And obviously, all these organs that have or can have fibrosis affecting them, the process, broadly speaking, is very similar. So there's some kind of injury in the liver that can be something like a side effect due to a drug that's metabolized in a particular way that results in the buildup of a toxin. It could be viral infection, in the same way that similar things can occur in other organs, such as the kidney or the heart or the lungs. Anything that causes injury and inflammation causes the release of cytokines, growth factors, peptides in the circulation, all of which contribute to that inflammatory response that can lead to a scarring process that is useful in the short term because it maintains the integrity and the structure of an organ so it can heal after that insult is removed. But if that injury or inflammation continues over a prolonged period of time, that healing or regenerative response never occurs. So you end up with scarring or cirrhosis. And as I say, depending on the organ that's affected, that will have different symptoms for the patient. But ultimately, it will lead to obviously clinical symptoms and in some cases death. And there are no drugs presently approved that can actually reverse or inhibit fibrosis. So it's obviously an area of interest to try and develop those treatments that might allow us to treat these conditions, rather than make lifestyle interventions or provide organ transplants as kind of the only kind of solution, which obviously is not by no means perfect or applicable to every patient. And this cartoon that I've borrowed from these two authors is kind of giving a heads up a view of what that process of fibrosis is like. So if you think, it doesn't really matter which organ you're talking about at first, in very general terms, you have all these different cell types that are involved in the process of fibrosis. One particular cell type is the pericyte, and in the liver, we tend to refer to these as hepatic stellate cells. And different scientists will argue about how similar or different pericyte populations are in different organs. But generally speaking, these resident pericytes are found closely associated with the capillaries in whichever organ you're talking about. And in response to injury, they will become activated into cells that express things like platelet-derived growth factor receptor beta and transcription factors like GLI1, which these particular authors were interested in, which is part of the hedgehog signal pathway, which you may have heard of in development and also is involved in some types of cancers. And when they activate, these cells adopt a fibroblast phenotype. So it doesn't matter which organ you're talking about, during that fibrotic process, you will have cell types that exhibit similar characteristics. One of which is that they express a protein called alpha smooth muscle actin. So that goes alongside these cells being contractile. They exhibit that particular phenotype, which they wouldn't in a healthy organ. And they also deposit, as I said, these proteins that make up the fibrotic scar. Depending on the organ, there might be different populations of resident fibroblasts that might also contribute to the fibrotic response. And in some cases, there may also be examples of cells migrating into the tissue to contribute to that response. But in the liver, at least, I'm primarily interested in a resident population of cells called hepatic stellate cells that exhibit this phenotypic switch, which as a basic scientist is really interesting to research because it involves lots of chromatin remodeling and changes in transcription and gene expression, which we're beginning to understand a lot better. But if we could understand it even further, it might lead to developments that could identify new treatments, for example. And one particular transcription factor, so I've switched what the focus of this review article was, you could substitute your transcription factor of choice into this cartoon. In my case, my favorite transcriptional regulator is called yes-associated protein 1, or YAP1. And this is a transcriptional regulator that we know is important in organ growth and development, and is also mechanically sensitive, so it can be activated by mechanical cues. So there's obviously, it's a fairly obvious idea to think that if you have a scarring process which changes the mechanical properties of a tissue, there's going to be an involvement of mechanosignaling pathways in that process. As the cells respond to the altered, scarred environment, that's going to switch on different pathways of gene expression and the expression of different proteins. So could interrogating or investigating these pathways reveal novel targets for new therapies for fibrotic disorders or diseases. And we investigated this, as I say, in the context of liver disease. And so if you think about a healthy liver, relatively speaking, it's quite a soft cellular environment. The cells are not exposed to a great deal of, or not a very high mechanical stiffness. But as the tissue becomes scarred and as injury occurs, the mechanical stiffness increases. And so you have cells that respond to that change in the mechanical environment, and they change their expression of things like the S-associated protein that I mentioned, which in a soft or healthy tissue is found in the cytoplasm of these cells. But once they become exposed to a high mechanical stiffness, which we can do in the lab on hydrogel substrates, the S-associated protein, YAP1, becomes nuclear expressed, where it can regulate target gene expression. And so this is something that we are interested in. We observed this occurring. So the question is, how does YAP1 becoming nuclear localized and switched on in response to mechanical cues, how might that contribute to the fibrotic process? And does it have a role to play in driving the activation of these hepatic stellate cells, which we know are the bad guys in chronic liver injury because they make all that fibrotic scar protein? So again, to try and be a little bit interactive, if we switch back to my Menti presentation, so if you've lost that screen, just go back to menti.com again, and you should be able to use the same code to catch back up to where I'm up to. So I'll move on to the next slide, which is to get you to think a little bit about words associated with fibrosis based on my very rapid introduction to the subject just now. So what kind of words do you associate with fibrosis? Or phrases for that matter, that I've very briefly mentioned? Or are there any things that I haven't mentioned that maybe you would think of? So yeah, certainly in the context of liver, alcohol comes up a lot, but it's by no means the only cause of liver fibrosis. And surprisingly, perhaps it's not the leading cause. So certainly in the US and nations with other similar problems with obesity, one of the leading causes of liver transplant now is patients with metabolic syndrome and fatty liver disease. But yeah, definitely scar tissue. In the context of my course, definitely hepatic. I'm focusing very much on liver, but fibrosis can occur to any organ or any tissue. And it does result in scar tissue or scarring. So whenever you hear that word fibrosis, it probably just means you're listening to a scientist like myself talking about a process related to scarring, but rather than it being on the skin, it's of an internal organ. And yeah, definitely stellate cells. They're my particular favorite. So I'll switch gears again. Thank you for your answers. I'll switch back to my slides again, which hopefully is working okay. So the question now is, what is my research right now? What am I up to at the moment? And so I was fortunate, when was this? Last summer, to get some funding from a charity in the UK called Guts UK, whose remit is to fund or to pump prime research around the digestive system. But that also encompasses the liver and also, in my case, fibrosis research. So they funded a project of mine that's still ongoing, looking at whether we could block any of the mechanosensitive pro-fibrotic YAP1 pathways that we'd identified as a means of slowing or reversing fibrotic disease. And this is, I guess, a pump priming project. So it's very much looking in vitro in our stellate cell models to see if we can find new pathways that YAP1 regulates, and whether we could inhibit any of those as a way to reverse or slow down their activation in our cell culture models. And so the way of thinking about this is, we know some kind of insult results in activation of these stellates, and we can model that in a dish by growing the cells on a stiffened substrate, and then look at what happens. And then we can obviously see that YAP1 becomes nuclear localized and activates target genes. So we can try and identify those gene targets and figure out what they might do in terms of maintaining stellate cells in the active phenotype, or even driving activation of more stellate cell populations. So we're very much interested in this question of how it is that YAP1 contributes to the fibrotic response as a result of mechanical cues. And so we have a list of different targets that we're interested in. Some of these are long non-coding RNAs, which are, as their name suggests, they are RNA molecules that are both long, so they're defined by the length and the nucleotide number, and the fact that as far as anyone can tell, they don't actually code for protein. So they're expressed in the cell, and they have some function, but it's not related to being translated into a protein. I also have some data around that hedgehog, a different hedgehog transcription factor called GLE2. And we're looking to test novel inhibitors of YAP1 to see what effect that has on the expression of these downstream targets, and whether that also inhibits or reduces the stellate cell activation. And we're also hoping to use YAP1 reporters to look at that as a readout as well, to see if we can use that to screen perhaps new compounds that target YAP1 or related molecules. The other question, so taking a step back, is how is it that YAP1 is initially switched on in the first place? We know it responds to mechanical stiffness, but we don't really know a lot about how that process occurs. So it's widely known. This is not something that it's just me that's working on this. Many labs around the world have observed this. It's been published widely by big labs in Italy in particular. And the question is, what's going on between the cell's external environment and the cytoplasm in the nucleus that allows or triggers the translocation of YAP1 into the nucleus, where it can drive gene expression. And so this work has been done by a PDRA, a postdoctoral researcher called Dina, who worked with me until recently, to investigate whether Enigma family proteins were involved in this process. And so this is just a quick run through of some of her data. So we hypothesized that Enigma proteins might be acting as a bridge, or as the things that allow YAP1 to translocate to the nucleus in response to those external mechanical cues. So the first step was to see if those proteins are expressed in our hepatic stellate cells. And so this is an immuno just showing in red, an Enigma family protein called PDLM5, co-localizing in a stellate cell, which is also positive for alpha SMA or APTA2, which is the marker of an activated stellate cell. And we can look at this by Western blot as well. So lysing the cells, taking the protein, running it on a gel, we can show that our favorite Enigma protein, PDLM5, is expressed with these blobs that we can detect using antibodies on our nitrocellulose membrane. And that is in cells that are also positive both for YAP1, but also our markers of fibrosis, ACTA2 and collagen 1. So at least in mouse stellate cells, which we have ready access to, we can show that Enigma proteins are expressed. We can also show that we're privileged that we sometimes have access to human liver tissue, and we can isolate stellate cells from those as well. And so again, we see in those particular cells that PDLM5 appears to be expressed by the red staining. So it's relevant in human cells. We think it's not just a phenomenon that occurs in our mouse cells. We can also use siRNA to target the expression of Enigma genes to see if that reduces the fibrotic phenotype of our stellate cells. And this is done in a cell line rather than primary cells. So these are immortalized human stellate cells. And this is just a graph showing qPCR data to look at what happens to the expression of both of two different Enigma genes and also the fibrotic genes that we're interested in. And if you knock down expression of either one of these, we seem to reduce the fibrotic phenotype. And then last of all, well, not last of all, but also to say that this Enigma protein expression also seems to be influenced by mechanical cues. So in our primary mouse cells, we can use our same model, culturing cells on a soft substrate to mimic healthy liver, or on a stiffened substrate to mimic the fibrotic phenotype. We can see how the distribution of these different proteins changes in response to those mechanical cues. And we think this might be involved in influencing YAP1 as well downstream. And then Dina went on to also show that PDLM5, our favorite Enigma protein, seems to physically interact with the S-associated protein 1. So it's in the right cell type. It seems to behave in the way that we hypothesized. And it also seems to interact physically with YAP1, which suggests it might be able to regulate it. So this is an immunoprecipitation. If you just use non-specific antibodies to try and pull out proteins from our lysed cells, you don't see a band in the immunoprecipitation for PDLM5. But if you pull out with a YAP1 antibody, you do get a band very faintly here, which we think is suggesting that PDLM5 interacts with YAP1. There's also a newer technique called a proximity ligation assay, which combines antibodies with DNA probes and then an amplification step using rolling circle amplification of DNA. And the amplification step is only possible if your probes that are labeled with DNA are in close enough proximity for that reaction to occur. And you only generate a nice red signal if that is the case. So if you don't see any red staining on your cells or on your tissue section, it suggests your proteins of interest or your whatever it is you've tried to label is too far apart for them to be interacting physically. Whereas if you do see amplification shown by the red signal, it suggests they are close together, which suggests they may be able to interact physically in your particular tissue or cell type. And Dean was able to show, at least in RLX2 cells, that this is the case. And so in conclusion, it looks like Enigma proteins are expressed by our fibrotic stellate cells and therefore may be able to regulate the mechanical activation of the S-associated protein 1, which might be relevant to understanding fibrosis and also hopefully down the line identifying novel targets for drugs or for biomarkers.
Thank you very much, Jim. That was very nice.
Hopefully I'm on time. Just quickly to say thank you to lots of people that I didn't mention properly, but Professor Karen Piper-Hanley, who provides access to some of the human tissue, my good friend Ricky, who's based in Spain for bioinformatics, Mark in Bristol, who gives us access to compounds, and then at MMU, I did mention Dina, so Dr. Abdul Motaleb, who sadly now left us to go to Birmingham, and then Steve and Tris, who are in the Centre for Bioscience at MAMA. But yeah, I will leave it there. If there is time for questions, I'm happy to take any. Otherwise, I will pipe down.
I'm afraid thank you very much for your talk. Very interesting, a whole new field, a whole new world for me at least. So thank you very much.
No problem.
So I'm gonna have to jump to the introduction for Dr. Jane Sarginson. She's gonna talk to us about pharmacogenetics. Dr. Sarginson completed a PhD in psychiatric genetics at Aberdeen University, and that was followed by a brief but memorable stint studying population genetics in sharks, complete with spear gun, before heading off to Stanford University to work in pharmacogenetics. She is currently a genetic lecturer at MMU, and she focuses on the role of genetics in common mental health disorders and their treatment. So another whole new world. Thank you, Jane, for being here. Welcome.
Thank you. And I'm pretty much saying now it's time for something completely different. So I'm going to try and just give you a bit of an overview of kind of pharmacogenetics and introduce you to some of the challenges that I've particularly been facing in the field of mental health, because obviously mental health is a little bit complicated. So probably at some point, all of us have gone to the doctor and we've asked them to treat something, and then we've been back 2 weeks later saying the treatment didn't work. We've got a bit of an issue at the moment that our drugs quite often they're only really effective in a small number of individuals. Other individuals will need to have the dose modified or will need a different drug. If you have a look at this slide here, you'll see that the individuals in blue are the ones who managed to get treated first time and had a good outcome off the drug. But these other drugs, and these are the top 10 highest grossing drugs in the US, the individuals in red weren't quite so lucky. So how can we think about improving that? Well, we have to consider a few different things. If you kind of have a group of people with the same diagnosis, they may not all have the same outcome. We hope that they'll have no toxic effects from the drug, so they won't have any side effects, and they will have a lot of beneficial effects. But there will be the unlucky few who not only don't have a beneficial effect from the drug, but have adverse effects. So, and some of these adverse effects can, in cases, be life-threatening. So we want to kind of try and identify these people so we don't put them on the drug in the first place. So traditionally, we've kind of done drug research with the one-size-fits-all outlook. So we do our research and try and get the right dose and the right treatment for the largest number of people within the population. And unfortunately, if you're not within that big group that have the positive effect, you kind of miss out a bit. So more and more now, we're moving towards personalized medicine, which is a big thing. And that's kind of tailoring the drug treatment to you as an individual. The aim is to deliver the right intervention to the right patient at the right time. We're not quite there yet, but we are getting a lot better at kind of like finding characteristics within these individuals that kind of are risk factors for a bad response to the drug. So we can kind of give you a bit more of a probability that this is going to be a good one for you and this drug isn't going to be a good drug for you. So the field of genetics within healthcare has changed massively within the last couple of decades. It's involved in every aspect of medicine nowadays. It's from diagnosis to prognosis. We have gene therapies, we have stem cell therapies. It's involved in drug development, and it's very big within cancer. But one of the ones that I'm most interested in is a specific field called pharmacogenetics or pharmacogenomics. And that is this targeting the right drug to the right person and getting it to them at the right time and in the right dose. And it also has an interesting sideline because this is becoming more common as well. And studies are beginning to kind of look at this early on. So it's getting introduced at the design phase as well as kind of the clinical trials phase. So we're beginning to kind of design the diagnostic tests we need to identify those individuals that will have the good response to the drug. At the same time, we are actually working on the drugs. The great success story in this has really been cancer treatment. So I'm just going to give you one example because this one is quite personal to me because it's recently a friend kind of came down with chronic myelogenous leukemia, and it around 65 to 90% of people with this condition, they have something called the Philadelphia chromosome, which is a translocation of the chromosome. So you get a bit of chromosome 22 on swapping with a bit of chromosome 9. And this does something really, really interesting. And this is a translocation that kind of occurs sometime during your life by accident. So it actually disrupts two genes, one being ABL, which is a tyrosine kinase involved in cell proliferation and differentiation and migration. And this gene is very, very tightly regulated. But when you swap it over and kind of stick it next to this BCR gene, you kind of change the regulation of that gene. So it's now regulated by the processes that would normally regulate the BCR gene. And it suddenly starts promoting cell proliferation and blocks adiposis. And it just isn't tightly regulated at all. So if we know individuals have this particular translocation in this cancer, we can treat them very specifically with tyrosine kinase inhibitors. And this has made a massive difference. It's like the 5-year survival rate has more than doubled from 31% in the early 1990s to over 70% for patients diagnosed between 2011 and 2017. And this is still going up. So another bit within this, which is actually my field more, is optimizing the dose and predicting adverse drug reactions. It's like, obviously, it's like your metabolism can affect this. It's how quickly you get rid of the drug from within your system can mean that either you're dropping out of the therapeutic window and losing the effect of the drug for periods of time, or it's building up within your system and you can get to the level where it becomes toxic and you start having side effects. There are lots of different processes involved in drug metabolism. So a common thing is to here referred to is ADME. So different processes affecting absorption, metabolism, the distribution and the excretion of the drug. And there are a lot of transporters involved in this, which is something I'm quite interested in. And we get a lot of this information from doing clinical trials. So we try and integrate the genetics into the clinical trials for the drug. But clinical trials, they're small, they're expensive, they're time consuming. And unless you specifically design them with pharmacogenetics in mind, it's very likely you'll only have a very small number of the minor homozygotes present within the trial. So it's a very, very challenging field, especially if you're like me and you work on antidepressants. So there are a lot of different factors that can affect drug response. It can be affected by your age, your sex, your mental health, your BMI, your kind of previous exposure to drugs, a lot of different comorbid conditions. And the genetics, the part of this that I'm interested in, is only a small part of this. So that's yet another challenge. And as we all know, psychiatric disorders are quite complicated. So one of the things that you really have to do within this is kind of think about picking your target. We have to kind of be sensible in what we look at and think about what the association could possibly be. That's why I'm particularly interested in the transporters. One that I'm very interested in is something called P-glycoprotein. So P-glycoprotein is quite interesting, and the area that I look at this within is actually antidepressants. And I'm going to quickly talk to you about a couple of studies that I've done over the years. I've been looking at this on and off for over a decade now, which makes me feel a little old. But depression, it's a common mental health disorder. There are 264 million people worldwide with this condition. It's an incredibly heterogeneous disorder with multiple interacting environmental and genetic risk factors. And it is more relevant at the moment because we're seeing rates going up with lockdown. It's like the UK survey that was done at the beginning of the year between January and March said that 1 in 5 adults in the UK had reported experiencing some form of depression in early 2021. And for those of you listening from abroad, that was a period we were in quite intense lockdown, especially here in Manchester. So antidepressants are a common treatment for depression, and the risks that come from giving somebody an antidepressant treatment that isn't effective for them, either in terms it doesn't help alleviate their symptoms, or it causes adverse side effects, are really quite high. It's like this can actually include things like suicide. as an increased risk. Okay, so just quickly have a look at this diagram, and this here shows some of the metabolic processes involved in breaking down the drug paroxetine and also kind of transporting it here into the brain. So as you can see here, ABCB1, which is actually the gene name for P-glycoprotein, is involved in the transport of paroxetine into the brain. And also here you can see that there are quite a number of cytochrome P450s, so liver enzymes that are involved in the breakdown of paroxetine. So this particular study is quite an interesting one, because in depression, especially in these individuals, they were quite severely depressed and of an older age range. So we couldn't leave these individuals untreated. So instead of doing a placebo-controlled trial, we compared two different drugs, one of them being paroxetine and the other being mirtazapine. So there's quite a lot of overlap in how mirtazapine is metabolized to paroxetine. But one of the things that is different is paroxetine is a substrate for P-glycoprotein and is transported by it, whereas mirtazapine isn't. So this gave us a really interesting opportunity where we could have people on two different drugs, but we still had something different that we could kind of compare between the two. So, as we said, P-glycoprotein is one of the biggest drug transporters to determine the uptake and efflux of a range of different drugs. So potentially anything we found was quite transferable between different drugs. Here you can see this is a survival analysis. So you can see that the people in red, it's like there, you can see these group of individuals who are carriers for the minor allele of this particular SNP that we're looking at. They respond quicker and in greater numbers to the drug, but it only makes a difference in paroxetine. So we're seeing pretty much no difference in mirtazapine, because it's like when this isn't actually a substrate of the transporter. So this result made a lot of sense to us. Okay, it fit with other results from around the time, although this highlights quite an interesting point about these kind of studies, because you can see quite a large spread here on the type of results you're seeing. So the paper, there they very specifically only used antidepressants that were substrates for P-glycoprotein. So it was quite a specific study, whereas these two results here, the STAR-D study and the general depression study, which are both much larger studies, they're both naturalistic studies. They use all sorts of different drugs at different doses, and they failed to find an association. So obviously we keep trying to find out more about this particular polymorphism, but we have a lot of challenges in doing this. This was a very interesting study because it was in individuals who were chronically treatment resistant and for depression. It's like they had gone through all sorts of different options. So this was an antidepressant trial with psychotherapy as well. And as you can see, it's an incredibly small trial. But even to get this number of people through the trial, this was a 3-site trial. And here we're seeing quite a good result, but it's only for 8 individuals. So it's like it became increasingly clear that we can't work in silos when we're doing this. We very much have to turn this into a global initiative. And one of the groups that have done that are the Clinical Pharmacogenetics Implementation Consortium. And one of the things that I'm doing with my research at the moment with other colleagues is trying to make sure that all the data we have is actually being fed into repositories, and people are able to kind of get access to this information. We can pull it together and try and get a bigger picture of what is going on with these specific genetic variants and how they're affecting individuals' drug response. If people are actually interested in this, I would suggest this is a very interesting website to go to. It actually has up-to-date information on dosing guidelines, information about drug metabolic pathways, as well as kind of suggestions of genetic variants that could influence them. Okay, I will just leave it there and give you an indication of how many different people are involved in some of this. It is, in fact, a very global initiative. Okay.
Thank you very much, Jane. Very interesting. I think that people that are not specialists in this field, we believe that one medication should work for everyone the same, you know, and it just fix everything as if it were magic, and we do not realize the difference.
Yes, I'd love it if that was the case, but unfortunately not very often.
I know. So I think for the time being, we don't have any questions on the chat. So we're going to start with our short break, and we'll be back at 25 past the hour. And we're going to listen to Dr. Bao Chang talking about stem cells for regenerative medicine. So please stay tuned, and we have a short break now. Thank you.